Well, hello, my name is Alan Garcia. I'm going to present to you the project that is Saccharomyces cerevisiae immobilization in graphene oxide for its use in microfluidic fuel cell. Now here I have the presentation outline. We have a brief introduction, some literature review, my methodology, the results, some of the data analysis, and my conclusions. So we're talking basically about a microbial fuel cell here, which is a device that generates energy out of a microorganism which consumes an organic substrate. Now, this process is made more efficient by using micro-sized channels. The main microbial fuel cells do not generate such a great power, so they cannot be compared to like solar energy or eolic energy, but by using micro-sized channels, this process can be made more efficient. Now, the development of a fuel cell can also be improved by making use of a paper, which I'm going to explain a little bit later. But well, the main focus of this project is to develop a microfluidic fuel cell based on a paper, on filtered paper. And also, if we want to implement this device in an everyday daily basis, we need to use a non-pathogenic microorganism. Now, small devices that we use every day, like, I don't know, pregnancy tests as well as others, they need small amounts of power and energy. And this energy is constantly provided by non-renewable energy sources, such as lithium-ion batteries. Now, this non-renewable energy sources could be in the near future replaced by our microfluidic fuel cells, such as the one that I'm seeing here. The main focus of this project is to develop a microfluidic fuel cell using this non-pathogenic microorganism, which is Saccharomyces cerevisiae, which is used for the development of bread, the development of beer, on a daily basis. It's been thoroughly studied by humanity, and we want to immobilize it on a graphene oxide substrate to consume different carbohydrates, in this case, glucose, fructose, maltose, and sucrose. Now, a brief literature review. Conventional microbial fuel cells, they function by having two chambers, the anode and the cathode. In the anode, we have our microorganism, which consumes organic substrate by oxidizing it. And in our cathode chamber, we have the oxygen reduction reaction, which takes place with the catholyte. As I said, the first microbial fuel cells were not so efficient. So this process was made more efficient by using microfluidic fuel cells and implementing our microorganism into the anode chamber of a small chamber that has some micro-sized channels. This obviously requires some special technologies such as sputtering and chemical vapor deposition. And the first microfluidic fuel cells contained the proton exchange membrane, which is necessary in a fuel cell to allow for the exchange of protons between the two chambers, the anode and the cathode chamber. Obviously, implementing this proton exchange membrane generates some extra losses that weren't required in these microfluidic fuel cells. So it was later discovered that these microfluidic fuel cells could be made membraneless. Why is this? Because our substances, they flow inside our chambers with a Reynolds number less than 200. And this means that we have a laminar flow and our two substances do not get mixed together. So this means that this allows some proton exchange between the two substances without allowing the substances to get mixed with each other. So it was discovered that this microfluidic fuel cells did not need any more a proton exchange membrane, which made it easier to fabricate them. And also it reduced the ohmic losses that were generated inside fuel cell. Now, to go even further about this, these microfluidic fuel cells were now developed based on paper, on filtered paper, as I said before. The previous microfluidic fuel cells that I've been talking about, they need an external pump mechanism so that the substances flow through them. But in this case, as we are using filtered paper and we're using the phenomenon of diffusion, we just need to deliver a small drop of our substance, of our organic substrate, and it is going to flow through the fusion among our paper. So this is going to also make it way cheaper to produce these microfluidic fuel cells. Some literature review. As I said, the carbohydrates that we're interested in consuming with our microfluidic fuel cell are glucose, fructose, maltose, and sucrose. Glucose and fructose are two of the most important carbohydrates in every single living organism, since we can get ATP from them, the basic energetic molecule. And well, in figure 9, I show a small schematic depicting the glycolytic pathway inside Saccharomyces cerevisiae yeast. Maltose, in this case, is a disaccharide made out of two molecules, which can be separated via an enzyme called maltase, which is reported in the literature is present in our yeast. Sucrose, just as maltose, is a disaccharide that is made out of two molecules of monosaccharides, which in this case are glucose and fructose, and it can also be separated via enzymatic by an enzyme called saccharase, which is also inside our yeast, Saccharomyces cerevisiae. In this project, we want to immobilize our yeast in graphene oxide because of its high properties, such as excellent conductivity, as well as all the functional groups that it has on it. As we can see in figure 10, it has aromatic islands, ketones, etc. This means that our yeast is going to better adhere into this surface, and it's going to allow for better direct electron transfer inside our fuel cell. Now, what do I mean by direct electron transfer? There are two different electron transfer mechanisms that can occur inside a microbial fuel cell. One is direct electron transfer, as I said before, in which our fuel cell makes direct contact with our electrode via a protein, such as a cytochrome. And there are also some cells that it's been reported that have nanowires, which are like super long proteins that make contact with a very far away electrode so that they can deliver the electrons that come out of their respiratory chain. Now, this is direct electron transfer. There is another one, which is mediated electron transfer, in which we have a molecule that can be constantly oxidized and reduced in our solution. This molecule travels to our microorganism. It gets reduced by the electrons that our microorganism produces, and then it travels into the electrode to get oxidized, deliver the electrons that our microorganism produce. Now, the hypothesis for this project is that our yeast, Saccharomyces cerevisiae, will generate electricity by consuming our 4 different carbohydrates. Our hypothesis number 2 is that graphene oxide will enhance this conductivity. And hypothesis number 3 is that paper will allow for an excellent base for our microfluidic fuel cells, since it is going to allow us to just deliver a small drop, as I said before, without any need for an external pump. Now, the methodology for this project was, we first developed 2 different catalytic inks. One is our anodic ink, which is the one that contains our yeast, Saccharomyces cerevisiae. As I said before, this yeast is the one that is going to be oxidizing our organic substrates. And in the cathodic ink, we will include some platinum supporting carbon, which is going to be in charge of the reaction of the oxygen reduction reaction. In phase 2, we prepared different carbohydrate solutions. As you can see, we prepared them in a range of 1, 2, 3, 5, 10, and 20 millimolar concentration, and in presence of a mediator, which in this case is potassium ferricyanide. And phase number 3, we develop our fuel cell. We use Toray paper, which is a carbon sheet as our electrodes. I'm going to explain this a little bit later as well. Each ink, and our anodic ink and our cathodic ink, obviously the anodic ink is going to be deployed onto the anode that we're going to be using in our fuel cell, and the cathodic ink is going to be deployed on the cathode that is going to be used in our fuel cell. And we use filter paper as the base for our fuel cell. And we used Walkman number 5 filter paper. And in phase 4, we characterize our fuel cell as well as our yeast via scanning electron microscopy. We also performed some cyclic voltammetry tests. We obtained open circuit potential, current amperometry, we obtained some discharge and polarization curves, as well as biochemical characterization with pH and temperature. Our cyclic glutamate setup was in a half cell, in which we had a working electrode made out of vitreous carbon, modified with our anodic, which is the one that contains our yeast Saccharomyces cerevisiae. We used a reference electrode that was made out of silver. That was made out of silver chloride, and we also had our counter electrode, which was made out of carbon. For our biochemical tests, the reactions are the ones that we show here in this equation. Our glucose in presence of oxygen and water reacting with glucose oxidase will generate gluconolactone as well as hydrogen peroxide. This hydrogen peroxide in presence of reduced dianisotine with peroxidase will generate oxidized dianisotine, which is going to shift in color, and we're going to record the shift in color in a spectrophotometer. So we can apply some equations and determine the enzymatic activity. For our fuel cell fabrication process, we have our filter paper sheet, Walkman number 5. We have our Torai paper, which are our electrodes. We deploy some of our anodic catalytic ink on the anode and some cathodic catalytic ink on the cathode. And then we put them together in a sandwich-like structure so that they don't have direct contact with each other, but they have direct contact with a solution that travels along the filter paper. Now, in our results, we have first scanning electron microscopy, in which, as we can see in figure 16, those little like spheres, they are our microorganism, our yeast, Saccharomyces cerevisiae, and it is supported on our carbon substrate. In figure 17, we can see the size, the length of our yeast, microorganisms, which is 5 micrometers, which is, according to the literature, the size of our yeast, Saccharomyces cerevisiae. In our cyclic photometry, we see 2 small peaks in the figure 18, which is the one with all blank, in absence of any of our carbohydrates. These little peaks could be due to the energetic reservoirs that our yeast has. And on figure 19, we can see those peaks are way amplified, like really amplified, as they are in presence of our 4 different carbohydrates of interest, which indicates that there is a reaction taking place. As we can see, most of them, if not all of them, and they have their peaks among the same voltage values. For open circuit potential, we can see here we obtain decent values for each of our 4 different carbohydrates. These values are seen better in these different graphs. In all of our different tests with the different carbohydrates, we obtained a value that was up to 0.75 volts, which is a decent value compared to the values reported in the literature. We also made some chronoparameter tests to test the stability of our fuel cell. We performed the test with a blank, with absence of our carbohydrates of interest. And we also performed the tests with our carbohydrates of interest. These values can be better seen here in these figures 27, 28, 29, and 30. We ran the tests with different concentrations of our carbohydrates. And we also performed some discharge and polarization curves. Here it is interesting to see that the highest values were obtained with glucose, but also with fructose, maltose, and sucrose, we also obtained some current densities and power densities that can be better seen here in these 4 figures here. For glucose, fructose, maltose, and sucrose. An interesting thing to observe here in these different graphs is that the power density as well as the current density increases in almost all of the cases as the concentration of our carbohydrates increases. And for our biochemical tests, we can see here we ran 2 different tests, one with a free yeast and one with an immobilized yeast. For the free yeast in the pH graph, we can see that the optimal pH was about 6, which is the optimal yeast reported for this yeast for this Saccharomyces cerevisiae. And we can see a very interesting result, which is that there is a shift in this optimal pH in 6.5 when it's immobilized. So this means that our system somehow changes the optimal pH for our system and makes it closer to the pH that well, we use in a neutral pH, which is present in most of solutions. And we can see also there is a temperature graph for the free and the immobilized yeast. In this case, we don't see any shift in the optimal temperature, which, well, it was too much to be expected to both of them to change. We can see here in data analysis, we have the results of the values obtained for our open circuit potential, our current density, and our plant density for our different concentrations in our different carbohydrates. The higher values that were obtained with glucose, but also the rest of the carbohydrates were able to be consumed and generate a decent amount of energy. So this means that our fuel cell can consume all of them and generate energy out of all of them. Now, in conclusions, our scanning electron microscopy analysis showed that our yeast, Saccharomyces cerevisiae, was immobilized onto our carbon substrate. Our cyclic voltammetry test showed that there is a reaction and oxidation process taking place for all of our 4 different carbohydrates. And the open circuit potentials, the highest value that was obtained was up to 0.93 volts. And our discharge and polarization curves showed values of current and power density, the highest values of about 0.3372 million per square centimeter and 0.056 milliwatts per square centimeter, respectively. Now, suggestions for future works on this topic are, well, we could perform some cyclic voltammetry tests with different scan rates to determine the reaction kinetics of our consumption of the organic substrates. We could also perform some electrochemical tests with our fuel cell in absence of a mediator to test the performance in this to test particularly the direct electron transfer of our microorganism and our microfluidic fuel cell. And suggestion number 3 is that we could test different systems for our anodic ink with different materials like carbon nanotubes. We used in this case graphene oxide, but we could also use carbon nanotubes to test its performance. We have here our references. And are there any questions? And thank you very much for your attention.
Muchas gracias, Alan García. Vamos a dar un pequeño espacio para ver si alguien tiene algún comentario, quiere hacer algún comentario, alguna pregunta del auditorio. De igual manera, Alan García, si quisieras ponernos tus datos de contacto.
Sí, claro.
Igual puedes compartirlo ahí en el chat o puedes ponerlo. Ah, perfecto, ahí está en el chat. Alan.garcia1135@gmail.com por si alguien desea ponerse en contacto con Alan para alguna colaboración o saber un poquito más del tema, lo puede hacer a través de este medio. Alan, te agradecemos mucho tu participación.